So good afternoon, everyone. It's my pleasure to welcome you on behalf of the Poultry Working Group supported by the PIG Commission on this webinar, Impact of Poultry-Related European Projects. So firstly, I will tell you some more about our Poultry Working Group. So we are supported by the PIG Commission and we have several aims or goals that we pursue. Firstly, we try to increase the visibility of poultry-related research at EAAP meetings. We also support scientific commissions on the EAAP meetings. We also organize joint undertakings with other organizations. And finally, we stimulate networking on the EAAP platform. If you like to read some more about our poultry work group, you can find some additional information online. Then who are we? I present to you my other colleagues in the poultry working group, Amelia from Germany, from the Hohenheim Center for Livestock Microbiome Research, myself and my colleague from the Flanders Research Institute for Agriculture, Fisheries and Food, Gurbut from the Research Institute for Farm Animal Biology in Germany, Katarzyna from the University of Nicolaus Copernicus Collegium Medicum in Poland, and Shali Turian from the Agricultural Research Organization Volcani Institute, Israel. Some of our members are also affiliated with WPSA. So our key activities the last year and also this year were the webinar that we organized last year on poultry science innovations and future perspectives in May. We also supported or co-organized 2 scientific sessions with other EAAP study commissions at the 47th annual EAAP meeting in Lyon. So today we are here to welcome you at our current webinar. And then this year we also co-organize a first joint EAAP WPSA session at the 57th EAAP annual meeting in Florence. So we hope to welcome there a lot of people. We also organize our own poultry work group session, and then we co-organize 3 sessions with other study commissions. So for the PIG Commission, we support in presentations on the heat stress. For the Nutrition Commission, we have some presentations on functional feed additives in poultry nutrition. And then for the Insect Commission, we support insects as food and feed. So for today, we first have some speakers, and then we like to finish with our roundtable discussion. And so we like to address which are the major issues defined in the projects, and then which are the solutions that are already suggested, and what are the further improvements or future perspectives that still need to be further explored. So this is the program for today. So we start with Ilias as a speaker, and then we follow for the next speakers, Daniela, Elisabetta, and Stefan. And then we finish with the roundtable discussion. So some practicalities. After each presentation, there is some time left for questions and answers. So everyone can raise their questions in the chat box, and then the chairs, me and my colleague Katarzyna, will read the questions, and then the speakers can answer. I also like to say that the webinar is recorded and will be available afterwards on the EAAP YouTube channel. So then I like to give the word to my colleague to announce the first speaker.
Thank you so much, Anatahia. The first, all our speakers are really distinguished, and we are very proud that you agreed to participate and share your insights in this webinar. The first speaker is Professor Ilias Kyriazakis, who specializes in the effects of animal management on their performance, ability to cope with challenges such as pathogens, and environmental impact. Professor Ilias Kyriazakis has worked with a variety of animal species, ranging from mice to cows. His recent work addresses effects of nutrition on the ability of animals to cope with pathogens, and use of alternative and homegrown feeds for livestock systems. and the development of methods to assess the environmental impact of local and global livestock. We welcome you, Ilias, with the presentation, suggesting discussion already in the title, collaborative research, a bottom-up or a top-down process. The floor is yours.
Be able now to see it. Okay.
Yes. Yeah.
Right. Good afternoon, everybody, and thank you very much for the introduction. I have been, during the past 5 years, I have been involved in 4 European Union funded projects, which focused on poultry. Of course, all projects were partly poultry, pigs. But Katarzyna asked me to speak about one of the projects, and interestingly, this is the oldest project in my repertoire in the last 5 years I have been involved in, and that project is PROHEALTH. PROHEALTH had the overall title, Sustainable Intensive Pig and Poultry Production. Obviously, I'm going to speak today only about the poultry component. As you can see, it has finished 4.5 years ago. Although it says that it started in 2013, in reality, it started in 2014, and it lasted for 5 years. It was, and I think it still is, the biggest project of its kind funded by the European Union, 12 million euros. It had 22 project partners, 10 of which were academic and 12 of which were industry. So there was quite a lot of interaction between stakeholders in the project. Now, I have taken directly from the call of the proposal what was the objective of the project. And the idea was to develop an understanding of the multifactorial dimension of animal pathologies linked to the intensification of production. So the term animal pathologies encompasses almost everything, like diseases, like disorders, metabolic disorders, for example, like skeletal abnormalities and everything like that. And the second objective was to develop, evaluate, and disseminate effective management and control strategies for these production diseases. Now, if you are wondering what are production diseases, again, from the call, production diseases are those who persist in intensive systems and whose prevalence of severity tends to increase with production level. So as you move from low to high input, the intensity of production diseases increases. And why are the production diseases important? They are because they have a great impact in both the European Union and worldwide in terms of system profitability, animal welfare, environmental impact, and product quality. They're actually the top reason that all these things are affected. The treatment and control is the major reason for the use of antibiotics in the food chain, and they're important in the context of sustainable intensification and EU food security. Please note that at the time the project was funded, 2013, one of the priorities of the European Union was sustainable intensification. The landscape now has changed to the Green Deal, but at the time that's what we were asked to address. So it was the funder who was calling the tune. What was it expected from the project? And that included the European Commission. And again, these are the 4 bullet points that were on the call, but they're not surprising. We were expected to have a better understanding of the various factors involved in pig and poultry production diseases. They wanted us to develop multifactorial control strategies that enhance product quality, animal health, and welfare. They wanted everything that we developed, the strategies, to have economic and social acceptability. So they wanted us to have an economic and social acceptability assessment of the strategies. And ultimately, and of course, I suppose this is what happens with every European project, they wanted to increase the competitiveness and resilience of EU pig and poultry systems. The emphasis today, as I said, is on poultry. So the project started from purely scientific starting point, and then you had quite a lot of social science to see what was first of all, the social impact of what you were finding and the economic impact. At that time, although we proposed it to the European Union, they were not interested in the environmental impact. This has become much more relevant as the years progressed. So now what I want to address now is what is essentially the title of my call. Who drives the research agenda, not only with this project, but any project will get funded by a variety of funders. And in my mind, and if you disagree, you can raise the point later, have left enough time for questions. One way of addressing the research topics is that the industry drives it. So the industry has a specific problem that requires a targeted solution. This is obviously a top-down approach. So they dictate the agenda, and I'm going to give you an example of what we did. There are two other ways of driving the research agenda, and I will give you examples for these as well. Okay, so let's take the first case, the top-down approach. So what the industry wanted us to do was to reassess the vitamin D requirements of broilers and layers against the legal limits within the European Union. The view at that time, at 2014, was that the dietary limits within the European Union for vitamin D inclusion in both broiler and layer diets were based on data before 2000. And these data, and obviously the limits propagated by EFSA, was that they did not consider the very rapid genetic changes in birth traits. 20 years, especially for broilers, is a very, very long time. So the view that was taken by the industry was that they may be associated with the increased occurrence of skeletal abnormalities. In other words, the levels of inclusion of vitamin D are low than what broilers and layers required. Now, what was quite peculiar in the legislation was that there was no distinction about the form of the vitamin D that can be added in the project. So the limit was basically about vitamin D3. And at that time, there was an increasing availability of one of the analogs of the D3. You are all familiar with it, the 25-hydroxy. And of course, you were able to add this analog that is much better metabolized by the birds, or utilized by the birds, I should say, and the issue became obsolete. So there was no worry anymore about this issue. The second example is that we had was to identify risk factors associated with the outcomes of production diseases in broilers and layer systems. And this was both a top-down and a bottom-up approach. The top-down was because the industry had extensive use of data, and they did not know what to do with it effectively. And the bottom-up was that the scientists are able effectively to develop protocols and methodologies on how to deal with the data, how to analyze it, and so on and so forth. And we used these data to identify risk factors for performance, mortality, and condemnation rates in broiler systems. And we did these three because that's what they were measuring, basically. So that was in their database. And here, this is a slightly difficult figure, but it just shows the probabilities of the different kind of outcomes and the probability of being affected by a variety of factors. If, for example, let's take the daily growth. Clearly, the daily growth is affected by the presence of disease, for example, but in return, how fast the birds were growing, they were defining whether they were arriving dead or alive at the abattoir, and the condemnation rate. The faster the birds were growing, the higher the condemnation rate. And you can see the other probabilities with the other outcomes. And finally, we have the third kind of driving a research agenda. This was the bottom-up. There was in the project, of course, the desire for novel markers associated with production diseases. And the scientists involved in the project had the desire to find novel markers for resistance to coccidiosis in broilers. And we did that. I will give you an example. We identified several genes that were differentially expressed during infection in coccidia. You can see, for example, here, the top panel is pro-inflammatory genes. The bottom panel is about the variety of CD4 and CD3. Actually, it's the other way around. The top is about CD4 and CD3 cells, and the bottom is pro-inflammatory genes. Of course, if you have the disease, the genes are expressed more, and you have a higher expression of pro-inflammatory genes, a higher expression of CD4 and CD3 chains, and you have a higher expression and maintenance of the interleukins. Now, what was quite interesting, and that's where the base for the marker discovery was, that all these were associated with faster recovery from coccidiosis. And they were also higher expressed in the slowly growing genotypes. We used slowly growing genotypes like the Ross Ranger, for example, against the fast genotypes, which were the Ross 308 and 608. And the question that we were asking, although we didn't answer it in the project, was, can these markers be used as markers of resistance? And interestingly, this was the continuation of the project. We got money from our national funder to answer this, which was basically a more fundamental question and less applied. So what did the project produce? What was its legacy? We obviously produced scientific information. We are in the business of producing that. But importantly, we influenced quite a lot European policy. We produced a number of policy briefs, and we had substantial presentations to policymakers, including one of the working groups of the European Parliament. We produced novel tools for the detection and control of production diseases. We produced an e-knowledge platform. At that stage, they were in their infancy. And we had a project website, and the industry actually decided to fund it for 2 more years. So the project website continued until March 2021. So just to conclude, the involvement in the project of multi-actors, it ensured that we strived to meet the needs of the variety of stakeholders which were the industry basically and the policy makers, but also the society, because as I said, we had social science involvement. And I'm pleased to say that the project was shortlisted for a Horizon 2020 Impact Award, but we were only shortlisted, unfortunately. Thank you. Oh.
Thank you so much for this presentation, for your speech. Although, yes, it is a past project, but it's you could say probably it's still living. The problems identified there are not fully resolved. Yes, some of them become obsolete, but of course, some of the results raised new questions, and this is just before continuation, my summary. I wish to also ask you, audience, to ask questions in the chat to Ilias for the aspects you are interested to or hearing in, or maybe some things you would like to have precised more. So the chat box is ready for you there. And my question would be, because of course you identified one project, national one, which was some continuation, yes, of the problems identified in the project. Do you know, because this was indeed historically the largest funded undertaking related to poultry research, other also species, but by European Commission. Do you know if there were other projects funded on national or EU levels that emerged somehow from results, or could be also listed as the continuation of your outcomes?
Yes, I'm not sure that I would call them continuation, but certainly they use the project as a springboard. For example, there is actually, I think it's still happening, a very nice project in Denmark. And the project in Denmark looked at the heel-bone damage in laying hens. And it used quite a lot of the information, for example, the risk factors we had identified. They used it as the basis for that project. So that was a springboard. In the pig component, there were at least two other projects that were used as the basis of these findings. One of them, in fact, in INRAE, where they were using the effects of the dirtiness of the environment on the productivity and resilience of animals. So yes, I would say that the project has been a springboard for a number of national projects.
Yes, so the legacy is living still. Yes, it's.
I hope so. Yes.
Yes, big success. We will then come back. There is a question from the audience just before coming to next presentation. One of the objectives of the ProHealth was to improve resilience of poultry production in Europe. Could you provide an overall assessment of the resilience on the current Europe poultry production and particularly poultry health?
Oh, well, I'm not sure I can answer that. Now, we have produced papers because we had economists in the project, which provided quantitative information about some of the treatments or some of the strategies we developed, what will be the consequences on the poultry sector. And in the broiler sector, for example, we found that some changes in the strategies we identified, they could increase the economic competitiveness of the broiler sector by 8%, which it was quite substantial. If people want to know a bit more details, please contact me and I can direct them to those papers. But yes, we have provided quantitative information.
Yes, good. Okay, thank you. We have to move on, but again, I say please stay. We will come back to the discussion in the panel discussion in the end, and I give the floor to Anna-Tahja to introduce the next speaker. Thank you so much.
Thank you. So I'm happy to announce the second speaker of today, Dr. Daniela Silva, who holds a degree in biochemistry and a PhD in biomedical sciences, and she is the chief scientific officer of ALS Life Sciences Portugal and Inventec Diagnostics within the biological sciences and biotechnology areas. Main research areas are focused on the use of bacteriophages to control bacterial infections and the application of molecular biology techniques for the diagnostic purposes in human health, animal health, and environmental areas. Dr. Silva, the floor is yours.
Hello all. Thank you for the nice introduction. I think I should share my screen, right? Let me try and put the presentation on.
Yes, indeed. Thank you.
So it's okay. So everyone can see the presentation. Okay?
Yes, we can. Thank you.
Okay. So I will talk to you a little bit about a EU-funded project that we had and ended in 2022, which was called PHAGOVET. And the purpose of, I don't know why it's not moving, right? And the purpose of this project was the use of bacteriophages to control E. coli infections and Salmonella infections in poultry. So there were one of the major drives of this project was the antibiotic resistance emerging in all fields, in human health and animal health, the recent constraints in the use of antibiotics in animal health, and particularly in production animals and birds. And considering these two infections, one of the major burdens for poultry producers and one of the major drivers of the use of antibiotherapy in agriculture. So what we wanted was to develop two products, one biocide and one technological feed additive, and this was the idea at the beginning of the project, to control E. coli and Salmonella. However, things are not always as simple as they seem to be. And what we found regarding E. coli was that E. coli is very diverse. It changes and mutates very swiftly. So encountering a fixed formulation that would be able to actually act on E. coli infections in poultry would be very, very difficult. So it would be like a tailored therapy or something like that. And so we have concentrated ourselves mostly in the Salmonella and go from there. So the consortium of this project was ALS as the leader of the consortium, and we were together with the University of Autónoma de Barcelona, SECAV, which is a cooperative of agriculture in Valencia, and VetWorks, a distributor and a consultancy company especially in poultry and in veterinary. But I've been talking about bacteriophages and the use of bacteriophages to control infections, but what actually are these bacteriophages for those who don't know? So bacteriophages are viruses that infect bacteria, and they infect bacteria in a very specific and very particular way. So, and they are not able to infect any other types of cells other than bacteria. So they are harmless to humans or to other animals. And this specificity of bacteriophages go to the species level, meaning that the bacteriophage that works against Salmonella will not work against E. coli or any other bacteria. And even within strains or within serotypes, the specificity is high. So one bacteriophage is not, it's not one bacteriophage works for all. One of the major advantages of the use of bacteriophages is that once the bacteriophage infects, it multiplies within the cell. So as long as we manage to put the bacteriophage in contact with the bacteria, it auto-replicates within the site of infection. And so you are always producing new bacteriophages to control the infection. So this, in theory, this is perfect. One thing that should be taken into account, because there are several types of bacteriophages, the lytic and the lysogenic. Lysogenic integrate the DNA into the DNA of the bacteria, and they can be hosts for antimicrobial resistance genes, toxins, whatever. So these lysogenic bacteriophages cannot be used for any type of therapy. So one of the major things is that only lytic bacteriophages, which have the life cycle that you see on the screen, are able to be used for therapy, where a bacteriophage specifically infects bacteria by recognition of some molecules within the cell wall. It injects its DNA in the bacteria and uses the bacterial replication machine to produce loads of copies of themselves. And then in the end, it results in the killing of the bacteria, because there are so many phages within this bacteria that the bacteria simply bursts. And there's the release of new bacteriophages that are on the site of infection or are near where the site where the bacteria are, able to replicate the cycle all over again. So, talked about bacteriophages, a little bit about Salmonella, because we really wanted to address Salmonella as a whole, not only considering obviously Salmonella typhimurium and Salmonella intrictidis, which are the major concerns because they are able to infect humans and produce disease, but also to control other serotypes of infection, Gallinarum and Pulorum, which cause disease actually in animals, and the other types of Salmonella that always make some constraints in terms of production, in terms of having the birds ready to go into the market. So we wanted to attack Salmonella in a transversal way. Obviously, the most interesting would be typhimurium and enteritidis, which are the major concerns, but we wanted to tackle all of these and taking these two approaches. First, looking at the bird and controlling the infection within the bird, but also controlling the infection within the environment. That's why we thought about that two-way strategy, the biocide and the feed additive that would be given to animals, and the biocide that would be used to spray on the installations, and the phages would remain on the beds and on the poultry houses, and somehow control and minimize the infection by Salmonella if any of the vectors like rodents or migratory birds actually transported any Salmonella bacteria into the poultry facilities. So what we have done a lot of work I will present here what I think are the sum up of the most interesting results and what at the end actually matters, which is, okay, the first part, which is the in vitro assessment of the Salmonella serovars that we are able to attack and to somehow the product would work, and the in vivo assays to show the efficacy of this product. So the PHAGOVET product that we were developing, it's composed of a cocktail of 4 phages targeting Salmonella. As I said, one phage will not be able to kill all Salmonellas, and even it's not serotype specific. So we actually need to find the correct combination, trying to have the broader action spectrum. So we've tested 271 Salmonella strains that were collected in all of the countries where the PHAGOVET has presence. So in Belgium, Benelux region, Portugal and Spain, and in Poland. We had 18 different serovars, and we tested loads of phages. And at the end, we decided that these 4 phages were the ones that together or in combination work better. So I have here the results. So the numbers in the table are the percentage of strains that were positively infected by each one of the phages. And as you can see, this cocktail not only tackles Salmonella typhimurium and Salmonella infectivis, but it's also able to attack, in fact, other types of Salmonella, allowing us to have a broader range of actions. So these are in vitro results, but as I said, there's one minor detail, which is not so minor, which is you actually need to put the phage in contact with the bacteria in order for the phage to work. So we need to somehow be able to deliver phages to the site of infection in order for the phage to encounter the Salmonella and infect and be able to kill it. So in terms of efficacy, we did a lot of trials. We did tolerance trials. We performed toxicities trials. So we know and we confirm that the suspension does not have any harm to the animals. They are happy. No significant differences were obtained, neither in terms of growth, water, or feed intake. So the phage solutions are completely harmless to the animal. So we really need to try and to prove the efficacy. And for this, we performed we performed several studies. I will show here an example of a short-term study and then a long-term study. So for this short-term study, we gave phages to the birds in day 0. Then at day 1, we infected with Salmonella typhimurium. And then we continue to give phages to the animals in day 2 and day 3. So our phage cocktail or our product. And then we took samples of local swabs and enumeration of Salmonella typhimurium in cecum samples. And actually, what we observed is that throughout the study, we had a reduction, although not very significant, of the counts of Salmonella in vehicle swabs throughout or in the cecum samples. It was more evident, this reduction comparing with the control. But we are here only in 1 to 2 logs reduction, meaning that maybe we need to improve our delivery of phages to the site of infection. But also we have here another important thing is that Salmonella, once infecting an animal, can reside in buffer cells. So once the animal is infected, there's always a resident population of Salmonella on the animals. So sometimes we don't see this so big shifts, but nevertheless, we were able to reduce. In a long-term study, we basically have an extra control, so the non-infected animals, a positive control, a treatment group. The assay was basically done the same way. So animals were fed with phages first, then were infected. They continue to receive phages, and then we made a reinfection, and then we continue to give phages to the animals. And this time we monitor local swabs, wood swabs, and secant samples. And here we actually see after 3 weeks and after 6 weeks, we see an actual reduction in the log number and the log counts of Salmonella, either in local swabs, in secant samples, and also in wood swab samples, which indicates that actually we are being able to control not only the Salmonella load within the animal, but also the expression of Salmonella into the environment, which was actually our purpose. So this is very nice, and from the beginning we have been talking either with EFSA and with ECHA to register this project as these products as a feed additive and a biocide. Many obstacles for both parts because there was no regulation, no guidelines, no regulatory framework to the register of phage-based products. And this was very hard because the rules changed and they asked every time more and more experiments, more and more things. We were not able, as it was our first goal, which would be at the end of this project to present a dossier for register for these 2 products. And then we changed strategy because only recently in January 2023, a guideline from the European Medicine Agency came out for a guideline on the quality, safety, and efficacy of veterinary medicinal products specifically designed for phage therapy. So finally, for the first time, there is a regulatory framework that enable us to register and to introduce into the market phage-based products. Although most of the work that we perform, thinking about registering this as a biocide or as a feed additive, will be able to be introduced in this EMA dossier. There is a lot more to do because it will be registered as a veterinary medical product. And so a lot more it's needed to be done, but this is really promising. We will not give up on PHAGOVET. So right now we are working on PHAGOVET 2.0, which will be the continuation of this project and the continuation of exploring the Salmonella phages against to control Salmonella infections in poultry, and taking now into account this guideline, and for the first time with a clear idea and a clear definition of exactly what assays we need to perform and what we need to prove in order to register and to put this product into the market. And that's it. Thank you for listening to me, and I'm open to any questions.
Thank you so much for this nice presentation. Up to now, I haven't seen any questions raised in the chat, if I'm correct. But I have a question for you. So we've been talking a lot, or we have heard a lot about Salmonella control, but the initial idea was also to work on the E. coli infections. Are there some future perspectives to proceed on that area of research?
Yes, we continue with the research with E. coli infections, but the thing is that E. coli changes so fast that the collection of E. coli that we had, and we have a big collection, so E. coli collections from 2013, 2015, 2019, 2021, and 2023. When you look at the lytic spectra, it changes completely.
Yeah.
So it's almost impossible to have like a fixed formulation, because you actually need to define a formulation of a group of phages that will compose your product, and then that's what you study. Although EMA opens and understands the specificity, so it opens the way that new phages can be introduced in a formulation once it's registered. So the idea is to tackle first Salmonella because it's very conserved. With these 4 phages, we have actually very good results and consistent throughout the time. For E. coli, the strategy is to have a phage library as large as possible. And I think it will have to be like a tailored therapy. So it would have to be like case to case. We can have like the flu vaccines. We can have the cocktail of the year, most probably to work. But in any infection, you will always need to confirm that that specific cocktail is infecting that specific E. coli, that it's on that region, on that farm. There's a lot of variability of what's going on in Portugal and Spain. They are more similar, but for instance, when you look on the profile that you find on Benelux or in Poland, they are completely different. So it would be more like a tailored therapy and not like a product itself. So we are focused more on Salmonella because as a first approach, we need to be able to. This would be the first phage-based product registered in Europe ever. So that there's no precedent to this. In FDA in the United States, yes, you have several phage-based products being used. But in Europe, no, there's no. So let's start with something that it's somehow fixed, and then, but yes, we continue.
Seems like a solid strategy indeed. Thank you so much. And now I give the word to Katarzyna to announce the next speaker, because I think we need to proceed.
Yes, it would be so much interesting to continue, but we need to move forward. We will come back to discussion. Thank you. And Elisabetta Giuffra, Dr. Elisabetta Giuffra, carries out animal genomics research across Italy, France, Sweden, and UK since 2011. Elisabeth is the research director at Paris-Saclay University, INRAE, AgroParisTech. So she pioneered international efforts to improve the functional annotation of farmed animal genomes with FANG and EuroFANG initiatives, and works in pig virus interactions using in vitro models and genomics approaches. And of course, Elisabetta coordinated the GENE-SWitCH project, and therefore the floor is yours, Elisabetta.
Thank you very much. I'm going to share. Can you see it? Correct?
Yes, it appeared.
Okay, so up. Voila. So thank you very much to the organizer for this invitation. And today, the idea is to tell you some hints about outcomes and perspectives of GENE-SWitCH, regulatory genome of swine and chicken, functional annotation during development. This project started in July 2019 and ended very recently, December of last year. So there is some premise to do about, before telling you what the project made. This has been really one of the FANG projects worldwide. FANG is the functional annotation of animal genomes. And why this? I mean, because in general, we know that genetic variants that in the genome affect disease and complex traits are typically enriched in non-coding regions of the genomes. And in time, the large human consortia for research, like ENCODE and many others, have really developed a large databases of data about the genome functionalities. That is something that you obtain by a large series of very complicated sequencing assays. And the idea is to obtain reference annotation maps that will help to identify which bits of the genomes are functional at a given time during development and so on. And FANG is an initiative that you find more at this website that actually promoted really an approach of open science to these topics with standardized procedures, coordination of the efforts of the community, infrastructures for data analysis, and so on. And all this because, of course, why we need functionally annotated genomes, because we want to decipher the genome to phenome link, so-called G2P or genotype to phenotype, and which is a bit, I mean, it's nicely represented in this very simple picture in the middle. Of course, you have the genomes, the genes, you have the environment, and in the middle you have phenotypes somehow determining themselves, because of course all this is the product of interactions of genome plus environment plus interactions genomes per environment. You can also include the microbiota in this, which is not exactly environment, but something different. It's a symbiotic reality we have. And this annotating genomes is needed to understand the link between genome and phenome and to be able to implement predictive biology. And this, of course, is in support of fundamental animal science that aims to link cell tissue and whole animal scale knowledge. I'm sorry, I have a problem of timing in the slides, but I missed. And this is important for dissecting emerging complex phenotypes and GPRE interactions. And very important for what is predictive biology for animal science, that is genomic selection. So linking trait-associated variation of a genome with function. And going deeper to understand these links, G2P, is necessary to identify causal variants, because sometimes genomic selection has limits. When, for example, you change your population, so you find a different genetic architecture and your scheme of genomic selection doesn't work anymore. Knowing the causal variants can lead you more far to use it more consistently across different populations, for example. Or it can help to reduce the search space and in general improve accuracy of genomic selection methods. So based on this premise, this consortium, a kind of small consortium, was put together that included a mixture of different partners, academic, including some FANG co-founders, including companies in animal breeding like Hendrix, associations for research and animal breeding, and as well a biotech company. So I let you simply read their logos here, and we will go back to this for the specific results I showed. So the goals of GENE-SWitCH, we can say two overarching goals we had. The one was to deliver underpinning knowledge on the pig and chicken genomes, because of course, everybody working in this field know that the genomes are never enough annotated. And when you follow an interesting gene or the top gene of your list, usually is an unknown gene. And to enable the translation of this knowledge to the pig and poultry sector. So it was quite an ambitious global aim. And it was developed in three interconnected aims. One was to develop, implement these functional annotations. And for this, of course, we couldn't do everything. We had also to coordinate with the remaining FANG community and the efforts, for example, in the US by our colleagues. And so we focused on development. We took a panel of 7 tissues and 3 developmental stages, 2 fetal or 2 embryonic for chicken, and 1 that was a newborn. The second aim was to develop methods and implement use of this functional annotation for precision animal breeding. And the third was to implement the standardization of data and processes and a strong action of dissemination and outreach. And of course, all these actions, all these aims were quite interconnected. About aim A, functional annotation, this picture shows schematically the kind of developmental stages we focused on: early organogenesis, late organogenesis, newborn hatched, and also adults, because the big advantage of having data with FANG standards is that you can use databases, the Tariba database then produced by other partners. And you see SWitch, that gave a name of the project, because of course, we had an interest to understand what are the genomic elements that switches between stages. And that could be particularly interesting also maybe for future insights in genomic selection, because the genomes during developmental stages is less known, less investigated until when we started. So we wanted to identify many different functional elements and especially produce a high quality and richly annotated genome map open to the community. And in fact, sorry, all the samples list, metadata, data, pipelines that we developed, primary analysis, it's all shared. It's open to the public in the DataFang.org website and under the terms of the Fort Lauderdale Agreement and Toronto Statement, which is the data policy agreement proposed in the FANG initiative. And this is to show that one main outcome of this first aim was a first ensemble regulatory build that, I mean, it's very famous ensemble, especially for the genomes of human and mouse and other key model species. But this on chicken and on pig was among the first for the genome of a vertebrate species that is different and still very important. This is described as a kind of review in the 2024 paper of Ensemble. And what you see in the figure is that as also another of the FANG projects, that was the one on fish species, AquaFang, we developed all these molecular assays that I showed in one of the first slides, and we actually contributed quite a lot to improve the annotations of the pig and the chicken genome, especially with the information on accessibility regions more recently with the conformation data capture data and also with histone marks and so on. But I don't want to go into technical details. In the aim B, we used functional annotation for precision animal breeding, and we took 2 different approaches. And this is probably, we go in what is of more interest here. So first question on the left was the approach in genomic selection. So can functional annotations enhance the prediction accuracy of breeding values in commercial populations? The question on the right was more an exploration project and was focused on epigenetics. I mean, can we detect the epigenetic effects of diet? This was only done in pig, so I will say instead some words about this. So the idea here was really to take the current genomic prediction models and try to improve them using the new annotations. And this was achieved by several means. So there was this development, but we generated fine-mapped QTL and DQTL data on dedicated cohorts of animals. And most importantly, these models were validated in large-scale commercial data. The main outputs that we had in genomic selections were, well, again, freely available resources like the Bayes RCO freely available in GitHub. Another important outcome, but here is just a flash, but you can actually find most of these already published, as I put in this slide, where that some very machine learning model that was used could better explain phenotypes for epistatic traits, which was a very interesting result. As I said, it's published, as well as it was demonstrated that gene expression explains considerable variance, but that depends on moment of sampling. And on QTL mapping, we had these studies, QTL in pigs and fine-mapped QTL in chicken. I will show one slide about that. And as I said, also the validation of these genomic prediction models in commercial populations. About chickens, there was a fine mapping study conducted by Uppsala University. And they actually working for many years on this topic. And for this, they used an advanced intercross lines that was produced from the famous Virginia chicken lines. You see them in, you see a picture in the left of the slide. So this is something that, I mean, I think there was like 60 years of divergent selection that led to have the high line and low line. So animals that have the same age, but are completely different in body size and growth rate. And using these cross lines generated from these populations, they characterized the genetic variants contributed to body weight in several individuals. And the idea was to overlap the EQTL study with GENE-SWitCH annotations to these regions. And they found, as I put below, about 42 regions harboring variation associated with body weight. They had also several outputs from overlaying of the annotations, like the genes and the non-coding RNA content was higher in these regions. And these data were provided to the study of commercial populations. And again, I give the part on the chicken. This empirical validation was made on 140,000 birds provided by Aviagen that were phenotyped for growth again. And here, what they highlighted was the potential of whole genome sequencing to enhance predictive performance beyond the 50K SNP that is normally used by industry. And they found a modest relative improvement in predictive accuracy when using exons from whole genome sequencing data. This is something, this modest improvement, which is kind of well known now, also from several other studies, trying to understand how annotations of a genome can improve the power of genomic selection. But there is apparently a considerable improvement when multiple layers of annotations are used. And this is going on now also for the chicken. by the same partners and others independently. And in GENE-SWitCH anyway, there was a very important result that was obtained in pigs, where combining annotations with the so-called Phages scores improved accuracy in pigs. And the link I put in these two slides is to send you to the original presentations of these people, where those of you who are specialized or interested in this kind of topics could find more information. Another aim of GENE-SWitCH, and I will go to accelerate a bit because I'm passing my time, was this standardization of data and processes, dissemination and outreach. And there were several activities, a lot on the standardization of all the processes, data, metadata, and so on. It was a very long validation, curation, archiving, etc. Where we had a lot of dissemination to the stakeholders, many kinds, and a lot for the breeding industry, of course. And there have been webinars, workshops organized, training, a lot of things that maybe some of you have seen or could find back. And I will say one word about these clustering activities with the other H2020 projects and the start of Eurofund, because these concern the perspectives, and so maybe you could consider as part of the future roadmap that this webinar aims to delineate. So these projects that you see, GENE-SWitCH, Bovrega, Aquafang, GERonimo, Rumigen, and Holoruminant, some of these projects are still ongoing, like GERonimo and Rumigen, and Holoruminant also, I think. They started essentially, at least the first three, they started together and had a number of common features. First, they really had the core activities, annotation of the genomes, but in different species of animals. They had this interest to use the annotations as well, like for the new models for genomic selections that we did. They were all co-developed, as usually many European projects are, together with European industry and associations, organizations, in general as full partners, and all made consistent efforts in dissemination, training, and networking. And what is interesting is these projects really work together, and they became the co-founder of a new project, which is not a research project, but is a project for the concept development of a future infrastructure for Europe. That would be for farmed animal genotype to phenotype research. And that actually will probably become larger and include more aspects strictly related to, anyway, the very universal concept, in my opinion, that is genotype to phenotype research. This is funded via Horizon, Europe InfraDev. And the idea of this project is to develop, consolidate, and optimize the European research infrastructure landscape, maintaining global leadership. We started in January last year. We will finish this phase next year, at the end of next year, coordinated by FBN Germany, in co-coordination with University of Edinburgh in the United Kingdom. And we hope we will establish the concept for an infrastructure to facilitate research and innovation for genotype to phenotype research in farmed animals and Europe. Did we match in GENE-SWitCH expected impacts? I think yes, I hope so. So I think we really contributed to deliver high quality reference annotation maps. We delivered and tested in commercial conditions new genomic prediction models, paved surely the way for follow-up investigation by ongoing projects like GERonimo and others. And we co-funded Eurofund, and then now it became a research infrastructure project that, as I said, provides a great opportunity for larger scope G2P research in Europe in the next decades. And I will finish by showing you the homepage of our website that is durable for some more years, and where you find all the outcomes of our final conference, including the videos of the presentations that I indicated in my slides. I end here, and thank you very much for your attention.
Thank you so much, Elisabetta, for this impressive presentation of the project's outcomes. In fact, we all probably, as you also suggested, that we could see and use your resources already, because probably I could say there are no researchers who have not heard about FANG or have not used the pipelines. Yes, so yes, the impact is just spread over our communities, and it's the amazing impact, yes, of the project. I am just trying to follow the questions from the audience, and have some comments that we are away a little bit behind with time. But it seems I would have a question to you, because all seems very clear, and coming to that discussion, yes, we do not have that much time. But in your opinion, what you find, or can you see any needs, or what could be needed to, or obstacles in generating even more impact? Because now the project seems to just flow and produce the sister and progeny projects, yes, but do you find it's just easygoing, or could you recognize some narrow or bottlenecks in the realization of your goals?
But one of the next steps, I think, is to really exploit at maximum whatever artificial intelligence offers, as it is already done in humans anyway, to really investigate and extract the information of a genome for making best use of it for genomic selection and maybe also other applications. I think that's one big part of the future. And then you have others. You have the possibility to use also different tools, in vitro systems, and so on. But anyway, I think that my view is that finally you want causality, you want to look for, and it's in the genome that you find it. It's a bit simplified, but I mean, what you need is knowledge of a genome, and of course, improved phenotypes. That's the other thing, which is a key thing. If you don't have improved phenotypes, if you don't define the new phenotype for the future, I mean, that are very complex. I mean, how an animal resists better to heat or drink less water, or if you don't define these phenotypes, you don't find the right answer. This is the limits, I see.
Yes, thank you. Thank you so much for the comments. And Collin now will present with the PPILOW project. Anne is the senior researcher at INRAE in France, specializing in thermoregulation, robustness, and early life management in chickens. And of course, coordinator of the PPILOW project, aiming at improving welfare in poultry and pig low input and organic production systems. And Anne, please feel welcome to share the presentation, and the floor is yours.
Thank you very much. And I would like to thank you, Professor Stadnica and Dr. Degrand, for this invitation. And I will present the PILOW project now, hoping it works well on your side. Is it okay with the screen?
Yes, everything looks good.
Okay, great. So PILOW is a European project. PILOW stands for Poultry and Pig Low Input and Organic Production Systems Welfare. And it proposes innovations for improving welfare in low input outdoor and organic poultry farms. So why is it so important to consider welfare in low input outdoor and organic farming systems? These systems are well known for the quality of the rearing practices and of the products, but there is still a diversity of practices throughout Europe, especially concerning organic systems, and still a need to improve animal welfare and limit mortality in relation to the outdoor run that provides the access to the possibility to express more behaviors than in confinement for the animals, but also challenges them. There are also ethical issues to tackle and a wish of practitioners and of the society for welfare improvement in these systems. So in this context, the objective of PILOW was to identify, test and evaluate welfare improving practices in taking into account environmental, economic and social impacts, including human well-being, according to the One Welfare concept. So here is the PILOW partnership. The project has been funded from 2019 until August this year and is coordinated by INRAE with 22 partners in 9 countries. And we are working together with 9 national practitioner groups, 4 dedicated to pigs and 5 to poultry. So these multi-actor groups are involved in proposing innovations for welfare improvement in both species, and especially concerning poultry for favoring positive behaviors, improving health and robustness, and also avoiding feather pecking in non-big trim laying hens, and also avoiding the elimination of layer male chicks. So we are associating the multi-actor groups to all steps of the project, from the inventory of practices and choice of levers to test for improving welfare, to the co-building of tools for welfare assessment on farm, but also for multi-criteria assessment of the levers for improvement of the welfare of the birds experimentally and studied on farm. And there are one welfare assessments made and business models built within the project. And we associate also the groups to on-farm training and change management until the implementation of innovations on farm. So what are the project outcomes and the impact generated? During this project, we have provided knowledge for practitioners, scientists and policymakers in the form of EAAP abstracts, but also reviews and on issues about welfare of these species, but also proposals of levers of improvement, and also studies on consumers and practitioner views on animal welfare. We are contributing to a better understanding of animal welfare and associated animal behavior, and especially in relation with the lack of use of the outdoor run, especially by broilers. So we have gathered knowledge on adaptability of poultry breeds to the outdoor area, and also on the individual variability on this item. We also contribute to a broader range of animal welfare management strategies and tools, especially with studies on management strategies to avoid feather pecking in non-big trim laying hens, and especially in considering the effects of green light in the setter hatcher, and also of providing insects to the pullets as chicks and on the outdoor run, considering also the management of the free range and the design of the barn in order to favor foraging and exploration behaviors and avoid fearfulness of the laying hens. We also propose management and nutrition strategies and techniques aiming at preventing diseases, parasitic risk, improving early life condition for chicken and laying hen robustness, especially dealing with incubation thermal conditions for improving resilience of the birds. Also, we are conducting field studies on on-farm hatching in organic and low input outdoor farms. And also we are testing fermented or treated feeds for preventing pathologies in laying hens. During this project, we have also tackled ethical issues and especially anticipated changes in national legislations concerning the alternatives to the elimination of layer male chicks. As you may know, in the layer strands, the males were and are still in some countries called at hatching because they are not very efficient to produce meat. In France and Germany, in 2023 and 2022, there have been national regulation to stop this practice in the hatcheries. And in PILOW from 2019, we have developed 2 strategies for avoiding the culling of layer male chicks. One is the study of new dual-purpose strains, both experimentally and on farms. So strains for having both eggs from the layers and meat from the males. And we have gathered multi-performance data and are building multi-criteria analysis and business models on their use. And we also develop an on-ovo sexing tool, so with a non-invasive method as early as possible during incubation. Considering the technical innovations proposed by PILOW, there has been the co-development of tools for welfare self-assessment in pigs and poultry, and especially concerning poultry, the extension of the EBIN app for smartphones that is a tool for welfare self-assessment by farmers or technicians on farm, and especially on organic and farms with an outdoor run. And during 2021, when the extension has been launched, until 2023, there have been 340 assessments for outdoor broilers and 390 for outdoor laying hens. And we are gathering the feedback from farmers. We have also provided a conceptual framework for sustainability assessment according to the One Welfare concept, so including an animal welfare pillar in the grids used for multi-criteria analysis in the project. And as I told earlier, there is also the provision of an in-ovo sexing tool. So with 2 parts in this task. One, identifying early markers of male and female embryos in egg contents and extra embryonic membranes at day 3 and 8 of incubation. And also by developing a new radiofrequency-based method for on-ovo sexing using millifluidic sensors. And this will be for the next generation of in-ovo sexing tools because the first one is already used in the French and German hatcheries. So here is an overview of the poultry production issues addressed within the project and at the level of the farm studies with the innovations implemented on farm. So we have tackled the issues of feather pecking, but also of the elimination of layer male chicks by the use of dual purpose genotypes and their evaluation. Also, we have tested on-farm hatching of slow-growing chicks in 2 countries, and we are testing alternatives to entailment ticks based on fermented and plant-based feeds. Besides this, we have longitudinal field studies for studying the impact of the long-term use of the welfare assessment applications on farm. So now for raising awareness among practitioners and training, we have organized technical days, training session, and making information for practitioners and policymakers. And we are now providing the results of our multi-criteria analysis and business models that we are currently finishing to build up. And we have also had dissemination actions towards different end users, practitioners, but also the public and policymakers. And there is the involvement of around 90 practitioners and members of associations within our PILOW multi-actor groups. So now for the further roadmap. From the project, more research and development is needed on dual purpose breeds, especially on the nutritional point of view, and also the product marketing and communication towards consumers and citizens. Concerning the early indicators of range use identified in broilers, we have now to discuss them as selection tools for practitioners and especially breeding companies. We would like also to test new incubation thermal programs in commercial hatcheries. There is also a need of refinement of tools provided by the project, such as the EAAP after practitioner feedback, and also refinement of in-house sexing tools for industrial use. Some questions have arose from the project, especially how to raise organic broiler breeders and pullets with outdoor access, and also how to conciliate welfare and biosecurity for the birds, especially when you have to confine them. And also the project will contribute in short and midterm to other projects and with links with other European projects, especially with the recently launched partnership on animal health and welfare that is coordinated by the University of Ghent, where some PILOW results could be useful, and also our results will be also of interest for the EU BroilerNet project. Complementary information is also needed on indicators that can be measured at slaughterhouse, because we were working on farm for PILOW, but there are also other welfare issues that have to be tackled, and this is done in another European project, which is the WISH project, coordinated by ILVO. And also the project provides some inputs for the INTACT project on product quality issued from some field studies from the PILOW project. And we will contribute to the knowledge for policymakers, considering further labeling and animal welfare policies that are currently discussed. So I thank you for your attention, and your questions will be very welcome about the PILOW project impacts.
Thank you very much, Anne. Thank you for the presentation and already clear indicating the future challenges and needs. So, do you have the question? is, have you already started some communication with the citizens about the changing chicken on the table? Yes, or how do you find, yes, any responses that you can share with us from that communication?
Yes, we have distributed surveys to the consumer citizens about the animal welfare solutions we could provide, especially the use of alternative breeds, or this is with this, we have gathered from some information from the consumers, and also we have made some communications through Terra Madre events, and also we have on some field studies, we have also the feedback from the consumers of, for example, dual-purpose breed meat directly sold by the farmers. So we have also their feedback on the way they consider the meat and also its quality. And in fact, we are within the multi-actor groups, we have members of civil society association, welfare associations for welfare improvement, and so they are much involved within the project, and also they will be part of in transferring information about this. And we have also had a meeting with the Eurogroup for animals at the level of the EU associations. So yes, we are in this dissemination part towards the society. But of course, the knowledge is still poor on, for example, the alternatives to the culling of layer male chicks, or even the way organic chickens are reared. People don't really know. Sometimes they ask, but do they go outside? This is just a basis of yeah. So we still have communication efforts to make towards citizens.
So, and your service, just to finalize these questions, are made in France or also in other European countries? These questionnaires are distributed and sent
yes,
in France mainly?
Yes, they were distributed in several countries, yes, European countries.
Thank you so much. We have another question emerging, but Anastasia, I am not sure if we have time, because we are really, really out, but we will leave it for the after the presentation to the end or to the next meeting. So I give now, thank you so much, Anne, again, really very much. And now Anastasia.
Yes, thank you. So I have the honor to announce our final speaker, Doctor in Veterinary Medicine, Stefan Gunnarsson, who is a senior lecturer in sustainable animal production at SLU. His scientific expertise focused on the relation between health and behavior in poultry and pigs, and he has been project leader for several research projects in poultry and pig health and both welfare, and he's currently coordinating the BroilerNet, a thematic network within the broiler sector funded by the European Commission. So Stefan, the floor is yours, and thank you in advance for your presentation.
Thank you, Anastasia. I will share my presentation here now. So, is this the proper one you see now?
Yes, indeed.
Yep, thank you.
Thank you.
So thank you very much for inviting me to this. I will present the BroilerNet project, and it is not an ordinary scientific project. It's funded by Horizon Europe Coordination and Support Action Money. So it's 50-50 with industry and academia. And we are 25 partners from Ireland to Greece and from Finland to Portugal. And we are quite active in social media. Here you have the QR code for our LinkedIn website, and you will I will give you them later. So be prepared with your cameras if you're interested, of course. And I got a very nice introduction from you. It's but what I would say here is it's my background as veterinarian, scientist, and teacher is maybe of some importance for coordinating a thematic network, but it's late in the afternoon. Experience from being farmer's son to a young, insecure young man and being organizing parties, etc., is also, I think, relevant for coordinating thematic networks. So the background here, I know that you're all within the business, but you know that the poultry meat will be the most widely consumed meat, and it's important in EU, and we are also importing a lot of poultry meat into EU. And there are challenges for having a sustainable global consumption, providing safe food that is of high quality, and the society has demand on a more sustainable production from environmental point of view, from animal welfare point of view, and from animal health point of view. And then also I would add to this that the European Commission wants to bring best practices to the farmer and see that good advices are not just locked into commercial companies. The BroilerNet project is a thematic network, and there have previously been innovation in laying hens. As you probably know, we have the 4D for F, data-driven dairy decision for farmers, the ongoing project of bovine with the beef sector, and EU pig is also going on at the moment. And then also the EAAP Agri operational groups are highly relevant within this atmosphere or sector. So the general aims of the BroilerNet project is to, of course, enhance the resilience and sustainability of European broiler sector. We want to bring more interaction between science and practice. And we also want to have a co-creation of ready-to-use innovative best practices. And for this, we need to mobilize all relevant actors in the broiler sector to work together. And the focus within this project is on the broiler farmers and the broiler farms. So we are not focusing on the breeding, hatching part, or the abattoir meat into the consumers. These are absolutely relevant, but we need to focus on something. And the focus is to have 3 themes, and that is environmental sustainability, animal welfare, and animal health management. And you will see like a blue thread through this presentation that they will come again. It's a way of organizing it. And here you have a complete picture of all the 25 logos of the 25 partners. And I think we stretched the network as much as we could with a budget of 3 million euros. And we're very happy that we could have the UK partners within the project as well. So specific objectives of this project is to establish broiler innovation network, bins as we call them. It shouldn't be associated to dust bins, but that is the abbreviation. And then these are created on national level. And then we want to create 3 thematic expert networks that is on a European level. And this is linked to our 3 themes. The tasks that these bins and tents are doing is to identify and assess the most urgent sector challenges. So within the scope of this project, there is not any research question given beforehand, because this is harvested or collected from the different countries. And with the processes, we will have ranking of challenges, then go back to the different countries to find good practices to solve these challenges, to perform evaluation and cost-benefit analysis of the good practices. And then when this process is at the end, we are making fact sheets and documentation and disseminating these best of the good practices to farmers and advisors. So the target group is always the broiler farmers and the broilers on the farms. So how are the bins working? They have a multi-actor approach. The bin facilitator is a person coming from the industry that is well aware and well acquainted with the broiler industry within the country. This facilitator is then gathering farmers and advisors and other people that want to join this, and they have, according to structured education, work as a facilitator where they make all actors to participate and to bring in all these sector challenges and try to define them. So here is a part which is mainly industry-led within the project. So here are pictures from bin meetings in Finland, in Slovenia. So you see that these are could be quite small groups from 8 to, I think the biggest one were about 40 persons. And they work according to a specific scheme. So a lot of this is to organize the challenges on the bird welfare, bird health and environmental sustainability. And here you can see post-it things on that are not organized. And then the groups also have to organize what is the most highly prioritized. We also create thematic expert networks across countries. And these experts, it's not necessarily scientific experts, it's experts from the industry. There are 3 groups according to the 3 themes, and we try to have an even distribution. So there are like 12 to 15 participants, well spread among the countries, participating partner countries. And they had to prioritize the top challenges and to select the best good practices based on a good practice evaluation tool that we have developed. And then in the end, they will choose the absolute best good practice, and that will be a person or an organization that will be awarded the BroilerNet champion. So here are the sector challenges for 2023. This is the first cycle. We will perform 2 more cycles. So you can see broiler welfare. It's the question of genetics breeding, farmer working training, stable indoor climate, health management, improving biosecurity, fighting against highly pathogenic avian influenza, reduction of antimicrobial use, and environmental sustainability, CO2 neutrality and environmental footprint, feed source and feed quality, energy management. So the TENS then asked the bins to collect the good practices from the different countries, and then they evaluated these good practices if they were technical, excellent, had an impact on the theme, easy to adopt, practical, had value added, co-benefits, minimal risk of unintended consequences, and if it's possible to generalize across Europe. And then they were prepared for a cost-benefit analysis. So the whole process here was that the TENS were getting the top 10 good practices within each theme. They selected 10 of them, voting via poll, and then they had to select one that was the top one, and that will be paid special attention. Those good practices that were awarded to the broiler champion, they also were evaluated through a cost-benefit analysis. that was based on farm surveys in all countries. Here we have had a challenge that farmers are not very happy of sharing information, but anyway, we got farm data of acceptable standards from all countries and tried to construct typical farm models, because these could be conventional farms, organic farms, or other types of farms. So there was an economic evaluation made of these best of the good practices. So the outcome of this first cycle is that we have had 28 BIN meetings across the 13 countries, more than 430 people within the sector have been involved in the project in this first round, and there was submitted 138 good practices. I would say that actually that was probably too many good practices, because not all of them were actually possible to score, that they would be possible to adapt to help the challenge or to mitigate the challenge. So going back to the sector challenges, I will not go through all the good practices we got, of course, because there were a lot of them. But I will give you an example of animal health management, that we in total got 45 good practices. And that was in improving biosecurity, fighting AI, reducing microbial use. And here is the top one that was selected by the bins, but also elected by the tens. And you can, in the next picture, you can see that improving biosecurity, we had dedicated small equipment house that there was a special color scheme used that was we got from France, or a group of farmers in France. Fighting avian influenza, there was a self-analysis of risk factors that we could get from Germany. And for reducing microbial late in the afternoon use water circulation, central heating with renewable heat from Finland. So here you have the QR code to our newly launched Broiler Knowledge Web. And the hub here consists of fact sheets, and we will add on some more. They are under construction, and these fact sheets will also be translated into the partner countries' languages in the partner countries. So far we have the foot health program from Sweden that was suggested in the animal welfare. We have a lighting equipment that could save energy and promote the light environment for the birds from Germany. In German, as you can see, and here you have an all-out no thinning method that is used in Finland, and this is in Finnish. So we will over time translate all these relevant good practices into the national languages, and they will be fully available through the Knowledge Hub. So the full outcome of this BroilerNet when we have finished in 2026 is that we will have 24 priority sector challenges that we have tried to find good practices, and that will be several good practices for all these. We will have 24 best of good practices, and we will have 24 champions. These are 3 per theme per cycle, and we have 3 cycles to go. And then we will perform the cost-benefit analysis of these good practices as well. So social media, we are available on X. You can follow us on X, and I think LinkedIn is probably better, as we have 119 followers on X, but we have more than 800 on LinkedIn. So that seems to be the social media that is attracting more professional people. So you have the QR codes here, and I will show them later again. I will soon be finished. I want to acknowledge the European Commission for funding this project. I would like to thank all that is participating in the BroilerNet team and working within this project. It's almost 500 people that have been actively participating. And in particular, the work package leader, Lisa, Paolo, Julia, Natalia, Monica, and Ingrid. And all the partner organizations for doing a good job. And here is the here are the QR codes again. This is the end. Thank you.
Thank you so much for this very energized presentation at the end of the afternoon. It was very nice to listen. Also, a lot of interesting information, including these QR codes. I see we have one question in the chat from Professor Kyriazakis, which is, what was the interaction, if any, of BroilerNet with the numerous poultry projects funded by the EU, say over the last decade?
I have a quick question there. As I was coordinator of Freebirds, which was a core organic project, and I was also participating in Innovation, although that was in laying hens, I would say that this first cycle we have been fully involved in creating and establishing the networks. It was so much more work for all the work package leader than we actually thought. So we have been, could you say that we have been quite turned into creating this network. So we will go further, and we have started with the first thing, and that is to contact all the poultry operational groups that are active, and we will continuing to approach relevant projects. We have the farm book, for example, that is ongoing, and we also, some are also participating in the Pathway EU project, which is on sustainability of animal production in general.
Okay, thank you. I hope I can still use or take some of your time to finalize this very interesting afternoon. So we have made some summarizing slides that we want to go through, and then we still have some questions for the speakers. So I will first give the word to Katarzyna, because your hand is raised.
Yes, I'm sorry.
No problem.
I did not even acknowledge that I raised my hand.
Okay.
That's a problem.
No problem.
No, but I wanted to read these questions. Yes, maybe this is a question to Daniela and Ilias. So maybe first, Daniela, yes, in animal nutrition and health industries, currently there seems to be a sort of hype about natural products, including certain plant compounds with relatively poor descriptions. What do you think about their use to control Salmonella, Escherichia coli, and coccidiosis? So, Daniela, would you share your opinion?
I think that multi-level approach, it's always best. So we should use every weapon that we have available in order to control this problem. So there are several natural compounds that show bactericidal activity, and I think they are promising, and if they should be used. The good thing is that with phages, they don't actually interfere, so you can use phages together with antibiotics if you want, together with any natural compounds. So whatever works, and I think that we really need, the main goal here is to find an alternative to the use of antibiotics, and we know that mostly they are forbidden in terms of animal production. So all alternatives available, I think if they work, we should be able to use it as long as we protect the animals and the animal welfare. All of them are compatible in principle.
Yes, thank you for the replying, Daniela. Because the author of the question was interested because it is very used in African countries, yes? So that was the major cause of the question. But Anatahia, as we are really close to ending the web, we wish to wrap up it with the initial idea. Maybe you would like to present our notes, which we've been taking before, based on a dissemination of your projects, but also during your speeches. So
for the ProHealth project, I noted that there's still some work to do on the environmental impact, but as also mentioned during the presentation, this was not one of the aims or the ideas at the start of the project, and it's really something that has evolved the last few years. So I don't know if there are still some additional comments or some additional improvements or further optimizations that can be put forward. But this is what we have extracted.
Can I say something? Because I have to go.
Yes,
unfortunately.
of course.
Yeah, you're absolutely right. The idea about environmental impact in all livestock systems has intensified in the last 5 years or so. In poultry, there is the misconception that the carbon footprint is an issue. Of course, it's not. But the important issue with poultry is actually in the nitrogen and phosphorus excretion. And in my view, this is where we should be focusing. And I note that several governments actually are now recognizing that in the Netherlands, in Denmark, in the UK, recognize that as a top priority. So this is something that we should be focusing.
Okay, thank you. For the PHAGOVET project, I noted down that towards the future, there's still some work on the regulatory part. As also mentioned, that this will be addressed in new projects and that special activities will be foreseen for stimulating the implementation, and that definitely for strategies towards E. coli, there is still some further optimization necessary, such as the library that needs to be further created. So I don't know if there are still some additional comments on this.
So we will continue with work that registered the product against Salmonella into the market, and still evaluating how we can move on with E. coli and which strategy would be better, taking into account what are the legal or regulatory constraints of phage therapy, which is very, very new. At least in Europe.
Thank you. Then for the GENE-SWitCH project, I think there is still some work, and which will be definitely addressed with the new facilities to expand the availability of genomic information, and then also how to translate this to the phenotype. So definitely some more understanding towards the environmental impact is also something that still needs some work, as also addressed in the other projects. And then I think
maybe you could shorten it a bit if you want.
Yes.
Yeah, I could actually send you an email just after and give you a suggestion. Maybe it's easier for me.
Yes, okay.
If you send me this paragraph, I can shorten it. Because, I mean, for example, you mentioned genome editing that was not really tackled in this project.
Mm-hmm.
And I also mentioned artificial intelligence, but this is within Eurofung, so you could shorten a bit.
Okay.
And also focusing on switches between stages that was done in the project is not much in the roadmap, in my opinion.
Okay.
If you
great.
want to do it now, I mean, I would suggest to remove explore precision genome editing
Yes, but we will send it afterwards via mail, and
Okay,
then
so if you send it, I will do it.
yeah, that's good.
I
It's
will do it
perfect.
better this way.
Yeah, no problem, no trouble.
Okay.
And then for the last two projects that were presented, for the PPILOW project, I think further dissemination of the practices is one of the major points that can be further worked on, because there's a lot of interesting information generated in the project. And I think already a lot of efforts have been made to spread the information, but I think we have still some work to do, as you also mentioned. So maybe this can also be a little bit shortened, but maybe the last part I want to stress that also towards welfare versus the biosecurity. Is it so that the parasites or the parasitic infections are there the most important part to focus on?
In fact, I mentioned it because it's also, you know, because of the avian influenza, you have to keep the birds indoors, whereas they have been outdoors for some of them before. And then you can have some behavioral problems, and they can start to have feather picking. And so here we have to take good care with enrichment and some ways to distract birds in order to avoid problems. There is also the possibility to add verandas, you know, a winter garden to the buildings. And in this case, some very first results from the project show that it could be beneficial for limiting feather pecking in case we have to keep the birds indoors. So it was more on this side of biosecurity I was talking about.
Okay, thank you. And then finalize for the BroilerNet project, we've written down widening broiler innovation network to implement and identified and verified good practices, sustainable global consumption and tackling societal demands, and then fighting against highly resilient avian influenza, and energy management and communication with farmers. Any additions or comments on this conclusion?
No, I think it's quite good conclusions. It's we will see in next cycle, because then we will harvest new challenges and we will try to find new good practices. Maybe we still have the same challenges. Who knows? But the challenge for future is to have this network going on, and a network has to have tasks for continuing. So I think that will be a challenge for the future. And that's why it's very important that we establish contact with other projects. So those new projects can fill the broiler network with tasks that the network could work with.
Yeah. Okay. Thank you. Yes. I believe that we can finalize within the academic quarter this webinar of this afternoon. I would like to thank all the speakers and also my colleagues from the Poultry Working Group for organizing this. And Katarzyna, I don't know if you still want to have some words to finalize.
Maybe just, I really would not like to have the final word, but I was just thinking about the common challenge for all the projects maybe, and maybe the thoughts that were suggested by Ilias also in the beginning. So what is it the driver for the projects? Is it a top-down or bottom-up initiatives, or combined, or bottom-up rather? In any of these, there is always, when we were listening to your speeches, visible struggle with the upcoming regulations, fast adapting to new regulations. Sometimes the problem that is aimed in the project is no longer valid or needs a change because the regulation changes. Yes, so there is a big and constant challenge with the policies, but also maybe overcoming limitations, especially with changing practices to when communicating with society, yes, and because in some countries also it's sometimes easier to perform some service, especially with farmers, yes, which are one of the major stakeholders of all these projects, and in some other even European countries it may be more difficult, still a very big challenge. So maybe this could be some also general challenges and maybe prospects or suggestions for involving more sociologists also and economists in the project. But it's also suggested by the European Commission itself, yes, for future. So yes, but I really don't like to have the last word. Please, Anatahia, feel welcome to finalize and close, but it's really, really a great pleasure to host all of you. Thank you.
And with this, I also would like to thank everyone that joined this webinar, and I hope you enjoyed this and that you have also some ideas for your own research. And with this, I think we can close this very nice webinar, and we hope to see a lot of you again this summer at the EAAP meeting.